Greetings, my name is Yesenia Castillo, and the title of my presentation is GluN3B Expression at Ventral Hippocampus to Infralimbic Cortex After Fear-Related Behavior. Post-traumatic stress disorder, or PTSD, is a mental condition characterized by the inability to inhibit fear. National Institute of Health defined it as such a disorder that some people develop after experiencing a shocking, scary, or dangerous event. The common causes of PTSD are rape, combat exposure, childhood neglect, or physical attack. During this traumatic event, the brain, specifically the hippocampus, collects data from our environment and send to amygdala to express fear. This kind of information can be vision, hearing, or smell. The PTSD patient uses this information as a cue or trigger to relieve this traumatic event. At the same time, infralimbic cortex modulates the amygdala to reduce the expression of the fear. In PTSD phenotype, in PTSD patient, this pathway is disrupted. That's why not all the persons that experience a traumatic event develop PTSD. Only one of each 9 develop PTSD. Previous results indicate that NMDA are the main player between the infralimbic hippocampus and the fear expression. Electrophysiological studies shows that NMDA current is reduced in our PTSD phenotype. These measurements are at this point where the AMPA current is on the baseline, so we isolate the NMDA current. All this information is based on male rats. So our hypothesis is that males and females will express less NMDA receptor on ventral hippocampus to infralimbic synapses after auditory fear condition. Now that we know that NMDA receptor is our main player, you can see here an NMDA receptor which is composed by different subunits. Here you can see the GluN1 and GluN2. However, we're gonna focus our project on GluN3B because it's the one that we have a lack of information, and also we don't have any pharmacological drugs that block or inhibit this subunit. Taking that in consideration, our question is, does fear learning alter GluN3B at hippocampus to infralimbic synapses? Our hypothesis is plasticity of ventral hippocampus to infralimbic cortex synapses contribute to fear-related behavior. First, we have to be sure that we only take in consideration hippocampus and infralimbic cortex. So we use an adeno-associated virus with enhanced yellow fluorescent protein, or EYFP, to track down the projections between one structure to another. First, we infuse the virus on the ventral hippocampus by stereothoracic surgery. Here you can see the site of the infusion. This green light is the virus. And here you can see the projections to infralimbic cortex. So basically we track down neuron to neuron until the infralimbic cortex. After the surgery, we wait 2 months for optimal expression of the virus. Then we go to the behavioral protocol. We randomly divided the rat in 3 different groups. The first one is our con group, which is our PTSD phenotype. In this group, we pair the shock with a tone. So every time that the rat hear the tone, they increase the expression of the fear. Also, we have our extinction group, which is our same group. We pair the tone with the shock, but then we have 2 sections of exposure therapy. So we expose the rat to the same tone that previously was a signal of dangers, now without the electrofusion. And lastly, we use our control group. We have the tone and the electrofusion, but it's not paired. After we run the behavioral protocol, we sacrifice the animal and take out a tissue punch in infralimbic cortex. Then we analyze the samples through fluorescent active cell sorting, or FACS. With FACS, we can measure the complexity and the size of these samples. We filter the size by 2 to 4 microns because it's the perfect size of the synaptosome. What is a synaptosome? First, the synaptosome is composed by postsynaptic terminal, in our case, frontal infralimbic cortex, and presynaptic terminal, which is hippocampus. That's why they express EYFP or yellow fluorescent protein. Now, how the machine works? Basically, the machine irradiates cell by cell and have these detectors that is able to determine if it's expressed the EYFP and if it's expressed our antibody. Here is the data from the machine. First, we isolate by size, you can see here. From this population only, we filter by the ones that is EYFP positive, that means that came from hippocampus. And you can see here, a rat without virus, you don't see almost any expression of this quadrant, that is the one that is positive for EYFP. And here you can compare with the EYFP rat, and you see the movement to the right that express that the machine is able to determine the specificity of the hippocampus projection. Now, we are focused on our protein of interest, which is GluN3B. Here is the expression of GluN3B and EYFP positive. You can see that in between female and males, and even treatments, doesn't see any significant difference. However, when you take in consideration all the sample of 3B expression with and without EYFP, you can see a significant difference between gender. Basically, between treatment, you don't see any significant difference. But when you compare the females with the males, you can see a trend. So this suggests that it's possible this change is sex dependent. Also, we take in consideration that some papers suggest that GluN3B is expressing in the presynaptic terminal. That means that it's expressing in the presynaptic terminal that came from hippocampus. To address this question, we take in consideration the smaller part of the synaptosome that contains only the presynaptic terminals that came from the hippocampus. Here you can see how we move our size to 1.5 to 2 and then graph them. So you can see that the expression of 3B is extremely low. This data suggests that 3B is a postsynaptic protein. And also we take in consideration the 3B overall with and without EYFP. So in conclusion, we can say, first, the data show a trend that auditory fear conditioning modify GluN3B expression in ventral hippocampus to infralimbic synaptosome in sex-dependent manner. 3B at hippocampus to infralimbic synaptosome is mostly in the larger EYFP positive synaptosome, suggesting that GluN3B is in the postsynaptic membrane. Our future directions are increase the number of animals per group so we can determine this sex dependence or if it's done. Thanks to my whole lab, especially to my PI, Dr. James Porter, and my techniques, Nani and Mari and Tata. And thank you to listen to me. And if you have questions, are more than welcome.